Thank you so much. Good morning, everyone, or good afternoon, depending on where you are. I'm going to start saying it's a great pleasure to be here today, and I'm going to explain you in the next few slides why I'm so privileged to be part of this webinar today. So I'm going to start, and I was asked in the first slide to show a little bit about my whole kind of academic trajectory. So I tried to put here a timeline that maybe is going to give you a little bit about my background. So as Alexandra mentioned, I was lucky enough to get all my training in Greece. That was in the University of Crete, where I completed my medical degree. And then I did a master's in neuroscience, followed by a PhD. And then it was a turning point that actually I was in one of the exchange students that we have between a program among the medical school in the University of Crete and the Boston University. So as a fifth or sixth medical student, I have the opportunity to do my neurology rotation at the Boston Medical Center. And that was for me the first time I was coming to the US. And I will say that that was the turning point. That was the time I say that I need to go to the US and continue with my training. And again, this was like primarily possible because of the motivation and the availability of the program that allowed me to come to the US. And I think this is something very important that we also would like to give back to many of the individuals that they would like to do the same thing. So I started residency at Mount Sinai in New York in 2008. Actually, the picture that you see here, that's my, it was like the first week I arrived in New York City. That's my official picture as a resident. And then after finished residency, I did a fellowship program, primarily in schizophrenia and genetics. And then I started being independent faculty within the Mount Sinai. I also have a role within the VA. That's the Veteran Affair, which is like the nationwide hospital here in the US that treats veterans. And throughout the years, I have been also getting different positions as a director. More recently, I have been having a position as the director for the Center for Disease Neurogenomics and also for a center within the VA that we do precision medicine. So I would like also to emphasize here the importance of the support of the HBA-USA, and there has been some amazing effort providing through this ASAP program. And I had the opportunity, two of the medical students, Pericles and Konstantina, were the recipient of this specific scholarship. And based on this mechanism, they were able to come to my lab many years ago and be able to spend 3 to 4 months just by getting some research experience. And actually, they were amazing. They were so good that after they went back to Greece, we actually offered them a research associate position, and they were able to come back to the lab and start working on very specific research programs. And the good thing is that both they were very interested after finishing the medical school to be able to enroll within a graduate program and do their PhD. And the good outcome is that both of them right now, they're graduate students within Mount Sinai, which is one of the most competitive graduate schools. And again, all this started because Pericles and Konstantina, they're amazing students, super motivated, and they also have lots of support from the HBA-USA to really be able to accomplish that. Now, in terms of the lab, if you know from New York City, we are actually over here as a Central Park, and you can see we're here on the top right, we're on the Upper East Side. So this is the Mount Sinai campus, it extends across multiple different blocks. And our lab is in a new building, it's called the Hess building, and we're actually on the 9th floor. So my office right now, when I'm looking outside the window, I can see right now here Madison Avenue. And this is the most recent picture from the lab. This is from, I believe, June or July 25. We have our annual retreat. And the work I'm going to be presenting today, it's only possible because we have put together an amazing team of multiple different individuals that really bring different expertise that coming together, we can really do what we call team science. So I'm going to start with a basic problem that I have experienced as a clinician. And as I said, I did residency. I'm a psychiatrist by training. And one thing that I really struggle still when I see patients is that when we have a new patient with mental illness, the way that we approach, we're going to do the clinical interview. we're going to end up giving a specific diagnosis to that patient, which is broad clinical diagnosis. And then based on the flavor of the clinical diagnosis, we have medication from 5 or 6 different categories, and then we're going to apply those medications and try to treat those patients. And one of the things that we see with the mental illness, but also with other disorders, we see that some patients, they do relatively okay. Some other patients, they don't really have any benefit. And for other people, no matter what we do, they don't really have very good response, a very good progress. Or they even develop side effect. And that's kind of an issue because one of the problems that we have is that we really do not customize our treatment or the diagnosis. So we apply the one-size-fits-all approach, which means that we give the medication and then we dilute the effect comparing to the one that might be more beneficial versus don't be beneficial. What we really want to be able to move is to be able to apply precision medicine and precision psychiatry. And here we try to take a very different approach. So what we would like, first of all, to do is we would like to utilize biological biomarkers or any other kind of genotypes or any other kind of molecular phenotypes that we can derive the patient and cluster them into meaningful subtypes. Then when we have that information, we are going to be in a much better position because this is biological driven subtypes to be able directly to address the biology underlying the etiology of those specific subtypes. So in that way, we can actually stratify the treatments that we give to different clusters of patients. And when this is done, then you will expect that the response is going to become more consistent and the overall therapeutic effect is going to be way stronger. Now, this is the promise of precision medicine, and of course, everybody nowadays, we all try to do this. It's a very challenging thing to do, especially in neuropsychiatric disorders, but this is where we actually try to really make a big effort because we think that even small increment on this front is really going to make a very big difference, right? Because we really want to make sure that we can treat patients in a more efficient and more effective way. So where do we start? Our approach is that many of those neuropsychiatric traits are extremely complex, but also they have significant heritability. And over the last 15 and 20 years, multiple large-scale genetic studies, or what we call genome-wide association studies, or very simply we call them GWAS, have really expanded to massive diverse data sets that they can actually statistically identify regions of the genome that they are associated with the risk of developing a disease. So I'm showing you here one example. Let me put my pointer so you can see better. So schizophrenia is a perfect example. It's an extremely heterogeneous and complex disorder. And as you can see, this was the GWAS that we published almost more than 10 years ago. And here we have about 35,000 cases and 47,000 controls. And based on that, we identify about 108 independent loci. So what you see over here, this is called the Manhattan plot. This is because most of the times you have really low buildings, like what you see in Manhattan, and then you have like the skyline that you have really tall buildings. And those tall buildings or those kind of association here is what we say statistically a significant locus that increase the risk of the disease. Now, what happened with those very complex disorders is that as you're increasing the sample size of your GWAS, then you find more independent loci and you have a better capture of the heritability of the disease. So as an example, 8 years later, we update that previous GWAS. Now we almost double the number of case and controls. And from 108, we went up to 287. More recently, our group led this specific analysis that we actually built on the previous freeze, which was back in 2022. And one thing that we did in this study for the first time is that we specifically increased diversity, meaning that we start doing genome-wide association study, not just looking European Caucasians, but also increasing dramatically the number of cases that we have with African ancestry. And of course, as you expect, with increasing sample size, and now this study almost brings together about 1 million patient controls, we have been reaching now about 400 independent loci. So this is great, and this is one of the power of the genetic studies, is that as you start increasing the sample size, and we do that more and more, we're going to finding more specific association within the genome that can really give us a clue about the etiology and the pathogenesis of specific disease. All right, this is great. Now the question is, what transformation have we done in the clinical practice? And the answer up to now is pretty much zero, meaning that after all these genome-wide and genetic studies, we haven't really changed anything in the way that we're treating patients. And we think that this is the start and the initiation of this effort, but there is still lots of steps that need to come together to really go from a statistical genetic association to be able to go to precision medicine or be able to do target discovery or even use that information for drug repurposing. So these are the stuff that I will show you today, and I will show you how we approach this very, very challenging problem. And one thing I would like to mention, because I'm not sure if everyone here on the call is familiar with those genetic studies, so I would like to give you a little bit of background why this is super challenging. So the first thing is that all these genome-wide association studies, they need very big, large cohorts because the effect size are very, very small. That's why we need to do like 1 million people to find statistically associations with those genetic variants, which means that even when we aggregate all this genetic risk score using a polygenic risk score approach, and we provide that score within a specific individual, as you can see over here, because the effect size is very small, it's very challenging to even say who's going to be a case versus control. And that's a very significant challenge. The second challenge is that most of this genetic variation, and it's not surprisingly, is going to affect the non-coding genome. And this is expected because only 1% of the DNA is going to translate to specific protein. So even from a statistical perspective and distribution perspective, you will expect that the majority of those loci are going to be part of the non-coding genome, which is more challenging to understand because we still have limited understanding of how the non-coding genome is going to regulate the output, which is going to be the translation of protein. Finally, for many of those significant loci, there is significant correlation between those genetic variants because of what we call the linkage disequilibrium. And as an outcome, you might have very extended regions that you have a statistical association, which under those regions, you might have multiple different genes and transcript, including like 5, 10, or 20, or even more genes. So that becomes very challenging, right? Because as you can see here in this example, it's very difficult to know if within this region, if I want to do a mechanistic study, I need to follow to gene 1, 2, 3, or 4. So this is one more challenge that we need to overcome in order to be able to go more to mechanism. So to address some of those limitations, 10 years ago, actually, yesterday I was in a meeting near Boston celebrating the 10th year anniversary of the Icahn Code. We actually established this kind of big consortium, which was primarily driven by NIH, and have been more than 100 investigators across more than 20 or 30 institutes within the US. And the goal of the Icahn Code is to actually be able to create a large, open-science collaborative framework that we try to understand the gene regulation of the human brain. That has actually multiple different fronts, from looking and taking brain tissue from cases with specific disease and controls, all the way to understanding how the genetics is going to affect those molecular markers in the human brain. And Icahn Code has been really doing a very good job to actually create very big resources and at the same time really do extensive studies that we actually communicate as a scientific outcome by coming all together, all the investigators, and creating what we call like a publication or a package of manuscripts. So we had one of those publication packages that we released back in 2018. That was the Icahn Code 1. And then last year, we also have the phase 2, which we released 14 papers that went to Science and other similar journals. And the one thing to say now is that the consortium has expanded since then. So right now we're working actually on the third release, which we hope to be ready for submission sometime next year. So let me give you one example out of all these different studies we have been doing as part of the Icahn Code of how we can actually use some of those resources to better understand the heritability of different diseases. And this is a study that was done by Biao Zheng. Biao was a postdoc at that time in my lab. Now he's instructor within my center. And what we do over here, we know that genetic variation is affecting gene expression. And we have been doing that for many years where we do transcriptome profiling using human brain tissue. And this is kind of design that we do what we call the expression quantitative trait loci. So as part of this study, we try to increase the diversity of the molecular profiling by also capturing the epigenome. So here we're using a specific assay, it's called ATAC-seq, that you can actually capture any region of the genome where the chromatin doesn't bound very tightly with the DNA. We call this the open chromatin regions, and usually these are the regions that different transcription factors that go and bind, and they're going to be regulating the gene expression. So most of the times you capture promoter, enhancer, and other type of regulatory elements. So we did that in the human brain by first separating the nuclei into broad neuronal population versus non-neuronal. We used FACS for that by using specific antibody that separate neuron versus non-neuron. And then we actually applied the ATAC-seq in those specific nuclei. So this is the largest ever epigenome analysis to our knowledge that has been done in the human brain. We generate 2,000 libraries from about 600 individuals, considering about 4 different brain regions. Now, having that information, we can then go at the genomic region and start creating a map of how the different promoters and enhancers they're going to be interacting with specific genes. And then we can now also start looking how the heritability and how genetic variance is going to be affecting those enhancer-promoter interactions. Having that, we can then integrate with all these genome-wide association studies and then be able to go from a genetic association of a genetic variant or a SNP to a very specific change that happens within those regulatory elements and with those specific genes. And as you can see here, for example, schizophrenia, about 70 of those loci, we have 270. So for about 70 loci, we found specific association with change in the gene expression. For about 35, we found that there's also epigenome and expression jointly. And we did that across many, many diseases. Now, this is a great step to really be able to do what we call fine mapping and try to do a dissection of the molecular mechanism. But we were also wondering, as you start doing all this analysis, do we have a way that we can at least functionally validate the way that we prioritize different genetic variants? So to do that, we actually turn on the massive parallel report assay, which is like a system that you can actually test thousands of different regulatory elements. So this is very similar to what we used to do back in the day using luciferase expression. So you have a specific cDNA construct, you actually put in a plasmid and then you throw within a cell, and then you see if the luciferase signal is going to go up or down. So we do something similar here. We can quantify with sequencing the expression of specific poly-A construct. But the nice thing about this is that we actually do that in specific cell types that they're more close to the human brain. So for example, in this study, we use excitatory neurons using NGN2 from stem cells. And the second thing is that we can test thousands of different constructs. And for this example, we test 20,000 different genetic variants, and we quantify how the effect of the allele, meaning that if you have the risk versus protective allele, is going to change the transcriptional outcome of those regulatory elements within the excitatory neurons. And as you can see here, after you do all this analysis, we found that genetic variants that we prioritize based on this pipeline by affecting the gene expression, these are EQTL, being within neuronal or glia epigenome QTL, or being within epigenetic regions, they are more likely to be functionally validated, which means that all the statistical in silico pipeline can really prioritize variants that then experimentally we can actually validate about the functional outcome. And here we have a nice example. So this is one region within, I'm sorry, I don't have the chromosome here, but it's very close to this specific gene, the RAP27B, which is a neuronal gene, very important for synaptic function. Here you have the significant locus from the GWAS. Here you see the signal coming from the expression QTL, epigenome QTL. Here we do statistical fine mapping. So from all these variants, we end up with one variant over here that statistically we say that this is the credible causal variant for this specific locus. and affect a specific regulatory element and this specific gene. And then when we did the MPRA, you can see that the G allele compared to the T increased the risk for disease, increased the expression of the gene, increased the epigenome readout of that specific regulatory region. And then based on the MPRA, we also have increased transcription activity. So that fits very nicely. And we think this is a really nice example of how we can demonstrate the power of this specific approach. Now, all these things are great. However, what I told you earlier was that the expression QTL that we have done primarily used bulk tissue, and even the epigenome that we did separation of neuron versus non-neuron really haven't addressed the heterogeneity and complexity of the different cell types. And this is something that really want to go. And nowadays there's like modern technologies that you can actually do single cell or even spatial analysis. Now, why is this important? Let's imagine that in disease framework, we're looking for changes only happening within the apples. If I do bulk tissue studies, I'm going to end up quantifying the molecular markers, almost like having a smoothie. And sometimes it's very hard to know if one smoothie versus the other has more or less apple. So by moving to single cell, we can now capture the specific fruits independently. Or even nowadays, we actually do spatial analysis that we can capture single cell, but we do that in situ. So we can even see how the different cells are organized and how they communicate with each other. So to do that, we would like to do this in a very large scale. And for that reason, we actually established last year the Genesis Consortium. This is a large-scale consortium supported by U24 from NIH, both NIA and NINDS. And the goal of this consortium is something super simple. Throughout multiple different consortia and efforts, we already have generated multiple single cell profiles within the human brain. But each study has been done independently. So there's not really common taxonomy for the single cell. There's not same naming about the cell population. And there's different pipelines that different individuals use to actually analyze those data. So what Genesis is doing is bringing all the data together, starting from raw. We actually propose everything jointly. We have the same taxonomy, and then we do the same thing for the whole genome sequence data. So we have a really nice harmonized genetic variation map that then we can actually use to do extended QTL analysis. So the goal of this consortium is to bring more than 10,000 single cell libraries and overall have more than 100 million single cell profiles coming from the human brain. So I can tell you that we are now in the middle of the year 2, and we already have reached 40 million single cell profiles. So we're very optimistic that within the next 2 years, we should be able to reach that goal. So in the next slide, I would like to show you one of the projects that we recently completed. It's called PsyKD, and it's actually one of the largest cohorts, single cell cohorts that we have within the Genesis. Now, for this project, the primary goal was to actually study the molecular mechanism of neuropsychiatric symptoms within cases with Alzheimer's disease, which is a very challenging area and is still not very well understood and very well studied. So to do that, we actually collect brain tissue from about 1,500 unique brain donors. This is a cross-disorder resource, meaning that we have not only Alzheimer's, but we have many other disorders, including cases with co-pathologies, but also with serious mental illness. We specifically aim to increase the diversity, meaning that we would like to have cases not only coming from European ancestry, but also be able to have admixed American and African ancestry. And we make sure that we also have an equal balance between males and females. For all these brains, we have very detailed neuropathology. We also have specific outcomes that measure the cognitive ability or performance of those individuals. And then we have also semi-quantitative assessment of neuropsychiatric symptoms that is very close to the time of death for those individuals. Now, all this brain tissue is coming within our center. And then we have a dedicated team of about 10 individuals that they actually work on generating this enormous single cell cohort. So the outcome was to actually profile 1,494 brain donors with single cell technology for gene expression. We generate almost about 3,000 libraries. And after you do all your QC, we end up with about 6.3 million single cell profiles. Now, as you do with any single cell analysis, you can actually use the molecular profiling of those single cells to organize those cells in different populations. And as you can see here very nicely, this is what we call the UMAP that include, here you should be able not to see, but here we include 6.3 million little dots. And as you can see here, the cells, they organize very nicely in broad different cell types, such as excitatory neurons, oligos, inhibitory neurons, and so on. And because we have really very high density of cell profiling, we can actually start defining even more specific cell types. So we did that using a hierarchical approach, and we end up going from 8 classes of broad cell type to 27 subclasses, all the way down to 65 subtypes, which you can see over here. For example, this is very important because, if you look here, we have excitatory neurons, but now by doing further taxonomy, we can separate those excitatory neurons into different subclasses, and each one has a very specific label coming from different cortical layers that they actually localize. And for a subset of those cases, we also have done very high resolution spatial transcriptomics. So when you embed those specific markers into the spatial omics, you can see very nicely the cytoarchitectural organization, meaning that over here is the surface of the brain. This is the cortex. This is white matter. Here you have the other part of the gyrus. And what you see over here is that in the top, in the layer 2 and 3, you have layer 2, 3 neurons, and then you follow this specific organization. So that really gives us a spatially deconvolute, high population scale single cell analysis that now we can actually use for doing multiple different applications. And just to put that in the context of other existing efforts, we also look the select gene. This is the biggest data set having single cell data across any effort, not just from brain, but including Human Cell Atlas and multiple different other efforts. And as you can see here, up to last year, and I think we checked also a few months ago, and this hasn't changed dramatically, you can see that our cohort with 6.3 million single cell profile is way bigger than many of the other efforts that have been done over there. And one very important thing is when you look for specific brain disorders, the majority of the existing single cell profiles directly have been profiling neurotypical controls, and we have very few ones that represent disease stages. So with this specific resource, we can dramatically increase also the number of single cell profiles that we have from specific disease cases. All right. So we have been using this resource, to start looking and dissecting the single cell gene expression across multiple different axes. One thing we have been very interesting is because we have a sub-cohort of about 300 neurotypical controls, starting from the age of 0 all the way to 100. We have started looking how development and aging is affecting gene expression. Then we have multiple different diseases, so we have been doing lots of disease analysis. And finally, all these individuals, they also have whole genome sequence data. So then we can also start looking how the effect of genetic variants, specifically on the gene expression. So I'm not going to have time to talk more about this specific project. I'm just going to give you a little bit of hint. But if you have interest, we have a nice page we have created for the PsychAD. If you go psych-ad.org, we have put there a list of more than a dozen manuscripts that you can also find as preprints. And all these manuscripts now are part of a publication package that it's either already accepted or soon to be accepted in Nature or Nature-like journals. So if you have interest, please follow this page, and you can find many of those resources. So I'm going to focus on something very specific here. And one way that we can connect genetic risk to biology is by conducting what we call transcriptome-wide association studies, or we call those TWAS. So the idea behind that is to be able to use genetic data to impute predicted gene expression in specific cell type, and then be able to test whether those predicted changes are associated with a trait of interest. So by performing TWAS at the single cell resolution, we can actually move beyond the bulk tissue signal and be able to identify which specific cell type or even subtypes are driving the genetic association of multiple complex traits. So this is one study that Sanand Venkatesh completed. He was a PhD in the group, and now he's a postdoc. And what Sanand did here, he integrated the genetic data with the single cell expression profiles from the PsyKD, which again include almost 1,500 individuals, and be able to do that across multiple different ancestries. And by doing that, he was able to impute gene expression at the level of the individual cell types. Now, one thing that is very, very important is that when you start doing all these studies, you start seeing that for given traits, it's important to have single cell analysis and resolution because not all the different cell types are equally important for a given trait. So what I'm trying to say here is, for example, if you have schizophrenia, you see that most of the cell type that seems to be important for schizophrenia heritability, they have to do with excitatory and inhibitory neurons, as well as also oligos. But on the other hand, if we go to Alzheimer, you're going to see more kind of reactive cell types, including microglia and immune cell type and astrocytes. So again, these are things that it's very important to capture using single cell profiling. Now, if we do exactly the same analysis and we do not use single cell, but we go with bulk tissue data, as you can see here, we have multiple different associations that you see here as blue as no, that we're going to be missing with the bulk tissue data. So in other words, by adding those single cell models, we can dramatically increase the power for finding statistical association linked to the heritability of different traits. Now, the other interesting thing is because we have single cell data, we can also study the pleiotropy of different traits, meaning we can see if a specific risk locus for, let's say, schizophrenia or Alzheimer is affecting specific genes and also understand the specific cell type. Now, this is very, very important because the next step after you do all this thing is you would like to go to more mechanistic evaluation. And you would like to know at least in which specific cell type you have an effect. Because if you try to do this, for example, in vitro, it's going to be very critical to know if you have to use, let's say, 2D models for microglia versus excitatory neurons. Or if you do that in mice, you'd like to know how you're going to knock down or affect the specific gene and in which specific cell type. So all this is very important information. So now we have created a huge resource that for every trait we can find how the specific trait associated with which specific gene in which specific cell type. Okay, let me show you one of the quick application of this cohort and how we can really go from something that we do discovery to something that might have like more clinical translation outcome. And I'm going to focus on suicide. And suicide is one of the leading causes of death. We have almost 1 million deaths each year worldwide. In the US, the rates have risen nearly about 30% since 2001, and this is really alarming. And this is especially important for the Veteran Affairs Administration because veterans, they're facing a much higher risk, especially women, where we see that the suicide mortality has increased more than twice as fast compared to the civilians. So what we did in this study, we combined all this genetic data. We combine with longitudinal electronic health record data that we have in more than 650,000 veterans that we have through the Million Veteran Program. So Million Veteran Program is a very big biobank. You have all the electronic health record data for those veterans that they come to the medical system for maybe over 20 years, including their blood work, their prescriptions, the medications, and then also we have genotype for those individuals. So what we do is based on the imputation single cell expression model that I showed you in the previous slide, we can then transfer those models within the MVP. So now we can impute predicted gene expression at the individual level and be able to directly link to the trait, such as the suicide risk. So the nice thing about this approach is that through this integration, we can bring together the statistical power of MVP, because it has 650,000 individuals with longitudinal data. But now we can actually combine that based on the cell type resolution that we can obtain from the single cell data sets. And that really can allow us to go from discovery in human postmortem tissues to insight that might be more relevant across a diverse clinical population. Now, within the Million Veteran Program, there's lots of phenotypic data. There's actually millions of phenotypic data within those individuals. And we can actually leverage this very rich clinical data to study suicide risk at scale. So, for example, here we have been using VA Suicide Prevention Application Network, with national death index and electronic health records, and we were able to phenotype almost 500,000 veterans. Among those, we identify about 30,000 suicide attempt cases alongside those with suicidal ideation or self-directed violence. And based on this phenotypic data and using some of the AI applications, we actually derive a stratified and ordinal phenotype that tell us about the severity of the suicidal behavior, meaning starting from no suicidal ideation, going to suicide attempt, either needed or not needed inpatient treatment, all the way to individuals that unfortunately they were able to complete suicide attempt. And now we actually use all this phenotype combined with the single cell data that we discussed, that I described earlier. So what do we get from here? We get this kind of results, which looks like a Manhattan plot, but this is not a Manhattan plot. Here we don't have a single nucleotide or single genetic variant, but each triangle here, it's actually the expression of specific gene within a specific cell type. And what you see here is that based on the color, these are different cell types, and based on the directionality, we can also infer whether the risk for suicide is actually increasing or decreasing the expression of a given gene. And that gives us a directionality, which is extremely important when we think about therapeutics. So the top hit that we found here, it's actually both in the bulk, but it's primarily driven by the excitatory neurons. And this is the gene, the HCRTR1, which is actually the receptor for orexin-1, which is actually very interesting because even when we use the imputed expression, we can find a very nice dose-response relationship with the suicide outcomes, meaning that veterans that they have no history of suicidality have very low imputed expression for that specific gene. And as you increase the severity of the suicide, we can see also that we have higher expression of that specific gene. And this is something very interesting that we're exploring right now, because this gradual increase suggests that orexin signaling contributes in a greater way to vulnerability for suicide, which make it both biologically meaningful and clinically actionable. And using this stratification, we can also potentially in the future, we can actually use that to stratify individuals and see whether specific individuals are going to be more likely or less likely to actually respond to specific therapeutic modalities. From an animal model, as you know, that we don't really have animal models for suicide. So here we use the chronic stress mouse model, which is a proxy for depression, anxiety, and suicide. And again, by looking transcriptomic across multiple different brain regions, we see that animals that they're actually susceptible and not resilient to stress, they also have elevation of that specific receptor, which again indicates that therapeutically you might want to actually antagonize and decrease the activity of that specific receptor. So to summarize, what we found from this study is that orexin-1 receptor is dramatically associated with risk for suicide in a very large cohort of veterans. And orexin in general is a very well-known pathway, and we already know that orexin-1 receptor, through multiple previous studies, including animal studies, we know it's very important in terms of regulation for reward, arousal, and emotion. And there's also another type of orexin receptor, which is type 2, and this is more involved in the sleep-wake stability. So our findings suggest that overactivation of type 1 receptor may increase vulnerability to stress and suicide risk. Therefore, if we go and we block that receptor, potentially we might be able to reduce the stress-driven signaling and be able to reduce the risk. So again, that was an idea that we were lucky enough because there's already FDA-approved medications that they bind to both receptor, both type 1 and type 2. And they actually clinically FDA approved for insomnia. Again, they have more activity on the type 2 receptor. But we're very interesting because there's significant value for really going here and really be able to develop selective antagonists of type 1 receptor, and also do the same for creating PET ligands, so you can actually monitor that in vivo in different individuals. But before we put all this resource and the effort, we're now doing a proof of concept phase 2 clinical trial by giving the non-selective antagonist. in specific individuals and veterans that they have increased suicide risk. And I can tell you from very early results, we already start seeing some exciting findings, but of course, we have to wait for the study to complete. All right, I would like to move to something else right now, and I'm going to try to finish my talk within the next 5 minutes. Orexin 1 is a great example. We were quite lucky to find that specific gene, but for most of the traits that we're looking, these are extremely complex and polygenic. So the likelihood of finding again single genes that we can really go after, it might be very low. So one thing we have been actually entertaining is how we can find a way that we can match polygenic perturbation that models the combined effect of multiple different genes. And our idea over here is that we would like to identify drugs across polygenic networks, not just a single target, that potentially they can actually use across multiple different diseases. So how does this work? So let's say that I have a specific disease change, right? So here you have disease genes, and you can see some genes that are actually going up and some genes that go down. Then I have a drug screening library where I have screened, let's say, thousands of different drugs, and I have the same molecular readout that I'm using to define the disease genes. So then what I'm trying to do, I'm trying to find the specific drug that is going to reverse the disease signature, and that's going to be the polygenic effect that's going to really try to reverse and potentially have a therapeutic effect. And the idea behind that is that we can actually do that by testing thousands of different compounds, even ones with unknown mechanism and function, or we can actually focus on very specific drugs that they already have passed the phase 1 clinical trials, so we already know the safety profile. So potentially if we find a beneficial effect for those drugs, these are going to be very easy to move to clinical trials and potentially be able to have something more into the clinical practice within 3 or 5 years. So let me show you one of the examples for one of the studies. And again, we have done this in multiple different studies because time is limited. I'm just going to show you a quick example over here. So this is a study we have been doing here at Mount Sinai with the Department of Neurosurgery. And what we're focusing here, it's the intracranial hemorrhage, or this is like one type of the strokes. This is not the thrombotic, this is the hemorrhagic stroke. It's less common, so it happens about 15 to 20%, but it's actually the worst outcome because when you have bleeding in your brain, usually you have very, very poor prognosis. So this individual, when they get acute stroke, they come here at Mount Sinai, and for many of those cases, they're going to be like brain surgery that they try to actually reduce the edema and remove the bleeding within the brain. And as the neurosurgeon is doing that, we're also getting biopsy from the human brain, which we actually use to isolate CD45 immune cells. These are the microglia and other adaptive immune cells. And then we did single cell profiling. Now, for all these individuals, we have longitudinal clinical data. So we know up to 6 months what happened to those individuals and how well or how bad they did. And one thing that we find is that when you look at the immune response, you actually have 2 different types of immune response. You have individuals that they end up having good outcome, and you see that most of the immune cells, they actually created this specific metacell cluster. And then you have individuals that have very poor outcome, including the one that they actually passed away, that they have this specific immune response within the microglia. So what then we try to do, we try to use this kind of polygenic drug perturbation and screen specific drugs that are going to convert this type of microglia, go more towards like this. And we end up with 2 drugs. And we actually test both drugs that were tested in vivo model. Here we're using mice that they have autologous infusion of blood. So this is a model for intracranial hemorrhage within the mice. And of course, when you do that, many of the mice, when you do like a neurological examination, they're including rotarod and other testing, you're going to see that they have really compromised movement and neurological function after you induce this specific stroke. But we find that after you give both medication, we have a statistical significant effect that protect those mice from this kind of neurological damage. And this is be done through modification of the microglia subtypes and really be able to convert some of the inflammatory microglia into the one that seems to be more protective. Now, this is great, but one of the limitations of our study is that we actually rely on drug screening data that have been generated previously. And when those teams generate those data, neuroscience and neurological disorders was not what they were actually targeting. So for that reason, many of us were using this data set, links CMAP, and there's not really much anything else besides that. And this is very worrisome because all these previous link CMAP data, they're using very generic cell lines. Mostly they're like cancer cell lines. They're using bulk gene expression, and it's not even RNA-seq. They use like a specific, it's like Luminex approach that you target almost 1,000 genes, and then you impute the rest of the genes. And when you look also those specific cells, after you give the drug, you don't have any specific phenotyping of the cell function. So that's very limited because we want to study the human brain, so we would like to have relevant cell types. We really believe that there's power of using single cell approaches, and we like to also have phenotypic that we can capture the cell structure and function of those cells after you actually expose those cells to specific chemical compounds. So in other words, we need to go from generic drug screens to more precision brain-focused discovery. So how do we do that? Because this is a very big operation, and if you want to do that, it has to be highly automated and very coordinated fashion. So for that, within our center, we actually develop a specific robotic system. We call it Wall-E, which was inspired by the movie because of the robot. And this is a system that can really host hundreds of different plates with cell cultures or other in vitro model. Everything is pretty much automated through robotic arms. And over here, we have a very specific liquid handler that can dispose microdosing of different drugs. And this is not based on pipetting, but it uses acoustic waves to really do very precise quantification. And then all the cells that can actually get different kind of cell phenotyping, including high context imaging that we do with an Opera Phoenix. But also we have single cell for all the cells, as well as doing electrophysiology and other cell phenotyping. So let me show you here one example. And this is a project we started with a big project contract that we got from the VA. You can see over here the robotic arm is moving. It's going to pick up a 384 well plate. So this is 384 well plate. In each well, we have a specific drug. We actually right now screening about 4,000 FDA or phase 1 drugs across different type of neurons. So we use here excitatory and inhibitory neurons that we actually derive from stem cells. So here you can see the plate with the drug coming within the echo liquid handler. It's going to actually reverse because then the next thing is to put the plate with the cells. So then you have attached those two different plates, and then you give the acoustic wave so that you can actually provide the microdosing of that specific drug. Follow that, we use cell painting, which you actually fix the cells. You use general dyes capturing broad structure of the cell. But then by using AI and CNN approaches, we can actually deconvolute thousands of different cell features that now we can actually use for finding effect of the different drug on the cell structure and function. And then everything that we do from those cells, they're also going to get single cell profiling. So I think we estimate about 50 million single cell profiling that we are going to generate through those screening approaches. So this is pretty much the pipeline. I think now we have the capacity to do about 1,000 drugs per week. So of course, this is the first effort is to create a reference drug screening map. But then we're going to be in a very good position when we have new patients that we like to bring, that we can actually start doing trial for each patient, which we think is going to be very beneficial for precision medicine approaches. All right, so I'm going to finish. I start my talk by showing genetics and then have like a gap here and how we can actually go to drug development or drug repurposing or even precision medicine. I think now that we have some amazing resources that we can actually utilize to at least attempt to achieve this very ambitious goal. In our case, we're using very large-scale biobank data. We have the power of the single cell analysis within a human brain from thousands of brain specimens, highly automated system that we can actually do large-scale drug screening analysis. And then, of course, everything I show you, we use very sophisticated AI algorithms to really be able to digest the scalability of all this data. So from today, I talk about the GWAS mechanism. So as we increasing the diversity and we do larger cohort, we're gonna find more risk loci, but still we need to have a functional outcome that requires single cell resolution. I show you some of the large-scale efforts that we are leading, including Icahn and Genesis, that we try to dissect and understand the molecular mechanisms underlying the heritability of different traits. A specific example within suicide, we found that orexin signaling might be important, and we want to follow up with that by doing an early clinical trial in individuals with high risk for suicide. But we also see that from the drug repurposing, we would like to come up with a pipeline that we can actually do that across multiple traits and also be able to apply that in real-world patients that we can actually use to select and identify medication that works the best for them. So overall, again, highly ambitious goal, but we think now we have some of the tools that we would like at least to attempt to go to more clinical translation. And again, all this work was supported and done by many individuals within the center, and of course, the many different collaborators across the world and within the US, as well as the funding from NIH, VA, and support from multiple different other consortia. So I'm going to stop here. Thank you so much again for the invitation. I know we're on the top of the hour, but I can stay a little bit longer. If there's a question, I'm happy to address.
Panos, thank you very much. This is an outstanding work and amazing presentation. Thank you. We have some questions. Alex, are you going to go over the questions from the attendees?
Yes, I can pick some of the questions. Regarding cell type annotation, which do you trust more, marker-based or reference-based methods, techniques?
I'm not sure what's the reference-based method versus the marker-based. So I will try to explain how we do this, and feel free if you can unmute and also ask the question. I'm happy to also have the discussion. So here as part of the BRAIN initiative, there have been multiple large-scale efforts, including things that we do from our group, to really be able to annotate and understand the molecular function and subtype of the different cell type within the brain. So for the analysis that I saw, we actually rely on many of the taxonomies and the nomenclature developed by the BRAIN initiative. And that's the way we have actually been able to identify and annotate those specific cell types. I think having the spatial data also help, because when we say that this is excitatory neuron of layer 2 and 3, then we can visually see that indeed it's over there. But one thing to keep in mind, and that's one of the limitations, gene expression is a readout, it's not the full multimodal capture of the cell. Of course, now we move more to multimodal, including epigenome and proteomics and other stuff. And there's also many other things that can be done, including electrophysiology, that we don't really capture here. So I expect that with more multimodal data, I will say that this kind of taxonomy should be expanded in the future and be able to capture not only the cortex, but capture more like subcortical areas. So I'm not sure if I answer your question, but feel free to drop more questions if maybe I didn't understand very well what you were asking.
I believe that was close to what he was asking, because I guess they're asking if Allen Institute and all of those different taxonomies that are out there, if they have been used. But always you are manually also try to modify those based on the data set, based on each data set. So I guess that's the answer that we're trying to find. The second question is similar to what I believe was answered right now from Dr. Roussos. It was easy. How are the classes annotated, which is what we answered right now. Is it through integration or special transcriptomics? So special transcriptomic, of course, help and mediate that characterization. Let me go to the next question. If there are any studies on the impact of infection, specifically herpes simplex virus type 1, in brain diseases such as dementia, as other studies or prior development?
Yeah, I know there has been multiple studies done in the past, and I'm not very familiar with the literature. I remember one paper published by Joel Dudley group here from Sinai. I think that was published in Neuron, if I remember, 2016. And the idea was to actually use bulk RNA-seq data and use the reads that they do not map on the human genome, but also map on specific viruses. And I think the one that they found was actually, I can't remember if it was herpes simplex virus type 1 or something else, but they found an association of increased viral load within those brains. Now, as you're doing all this analysis, especially when you're doing silico analysis, there's lots of technical variation that we need to consider. So I cannot say, and many other studies since then, they have actually provide additional evidence for the existence or no existence. So I cannot really comment if this is something true. But there's definitely nowadays lots of large-scale data. So I think it's going to be a good starting point to even start looking single-cell data and see if there's another evidence based on those data sets. And then, of course, you will need to do more elegant validation to really confirm or not confirm this hypothesis.
Yes, Stefan, you want to ask a question?
Yeah, okay, sorry about that. Yeah, that was very impressive, Pano. Really enjoyed it, even though it's not my field, actually. But however, a couple of things that I am working on. For example, your drug discovery efforts. So you have a massive amount of compounds that you try to screen for a certain phenotype. And I was just wondering, these have to cross the blood-brain barrier, and then a whole bunch of them, I'm sure they don't. And have you considered also to, among the hits, to try to prodrug them in order to make them cross the blood-brain barrier?
So we haven't done that yet. That's a great question. And to be honest, so for the very big drug screening project that we started a few months ago, I was quite surprised that nobody has really done a drug screening platform on cells that are important to human brain, like excitatory inhibitory neurons. And it's actually interesting because a few weeks ago, there was a really nice paper came out in Cell that they use omics in Alzheimer's disease, and they found 2 or 3 new drugs that then they test in mice. And if you read even the discussion, they say that we use CMAP and there's limitation because there's nothing about human brain cell type. So everybody understands this limitation. So to your point, I think we would like to start this by doing I think we use about 4,000 drugs that they are now phase 1 in FDA. Among those drugs, there's also the ones that we know that they're going to penetrate the blood-brain barrier. So that can be something that we do as a post hoc. However, I think this is a good starting point, but we have specific limitations, right? Because some of the drugs, especially drugs that we use in psychiatry, they need to have the first pass metabolic effect to be active. And for those, we don't really capture yet. So that's one problem. So we're aware of this. We're thinking that maybe we can actually do a follow-up study that for those drugs, we can actually get the active metabolites to be able to do that. But this is one of the limitations. Our goal is to be able, if there's the appropriate resources, to be able to do that across 50,000 compounds and really create a very big data set that, the whole scientific community is going to benefit from that.
Great. Do I have time for one more small question, Alexandre?
Yeah, I think Ivy was going to ask one.
Okay, go ahead. Go ahead, Ivy.
Thank you, Dr. Roussos. It was very interesting and informative. And although I have some scientific questions, because we're a little bit over time, and because I've seen a lot of junior scientists joining here, I think we can close this session with one question that a lot of people can benefit. So what were the challenges you faced when you had to transit from the Greek academia to US? And is there any critical advice you could give to all these junior scientists that they want to try to transit from the Greek academia or Greek environment to US? Thank you.
Yeah, my biggest challenge was to actually go from a scientific PhD level in Greece and come and start doing clinical training in the US. I will say that was the most scary thing, right? Because being doing research within a lab, and I did my PhD with Panos Roussos in University of Crete and also with Babis Savvaki that I see here on the call. Hi, Bobby. So it was great, to be isolated and do like your research. And then 1 week later, you come and now you're like in one of the most busy hospital in New York, in the US, and you're like in the psychiatric emergency department. You have to treat all these individual patients. So I would say that was kind of my biggest challenge. It was mostly to come to the US system, and I was first exposed as a clinician. But one thing is, there's lots of support over here. So even when you struggle a lot, I think motivation and commitment and finding the right mentors is going to be the way that you can actually overcome those challenges. And this is what happened in my case. In one way you get used, but then you have the chance to also meet amazing mentors. And by working very closely and creating actionable goals and reasonable goals, that's one thing that I need to keep in mind. And I say to every trainee I have in the lab, do something simple that you know is going to have an effect within a specific time. And when you do that, you can move to the other next thing. So I think that's kind of my biggest advice that I can give to trainees and individuals.
Thank you so much. That's really great.